Hi, everyone. My name is Theo Roth. I'm an assistant professor in pathology at Stanford University. I'm excited to be talking to you today about our lab's work on developing next generation genetic discovery tools for applications in human immune cell engineering. And so a central goal of what we focus on in the lab is trying to build technologies that can allow for dramatic reductions in both the cost and complexity of developing new, hopefully curative cellular medicines, and primarily for oncology applications. And the goals of this are not only to build drugs for current clinical indications, especially across solid tumors that we have poorly responded to immunotherapies and especially cellular therapies, but also to increase access to allow these to be a broader swath of the patient population, and then also to get curative drugs into patients sooner than traditional development timelines allow. And so today I'm going to focus on a couple of themes about how we've used genetic perturbation and kind of the development of new genetic software in a way to try to accelerate the development of curative cellular therapies in cancer. And so I'll talk today about kind of these four broad areas. First, how new genetic sequences can be integrated with endogenous genetic elements through targeted gene editing and how that offers unique advantages. How the amount of new genetic material that we can add into the human genome of a living human immune cell has been steadily increasing over the past 6 or 7 years. How we can build kind of what we call genetic software development environments or pooled screening methodologies to rapidly test large numbers of genetic constructs. And then finally, how we can increase the power of those constructs using kind of some new approaches beyond traditional gene knockout and overexpression based methods. And so I'll start with the basics of how integrating new genetic software into the endogenous genome can increase the effectiveness and potency of cellular medicines. And so most of the work that I will talk about today is all based on this magical ability that came around about a decade ago to integrate new genetic sequences into the genome at specific target sites using targetable RNA-guided nucleases, of which CRISPR-Cas9 is by far the most widely used. And so to introduce into primary human immune cells, just by using a simple physical delivery method of electroporation, large macromolecules from outside the cell can be introduced into the cell through the pores formed in the membrane of the primary immune cells by the electrical field that allows for both gene knockouts, but also if you introduce a DNA template along with your guided RNA-guided nuclease or Cas9 nuclease, you can also mediate gene knock-ins or introducing large sequences of new exogenous DNA to very defined sites of the genome. And so this is now growing to be work that we did a while ago now, but has now been actually these non-viral targeted integration approaches in primary T cells have now been used in a variety of clinical trials, both in academics and industry, by a number of companies. And so these have now been clinically validated. But there are traditional ways of introducing new genetic material into immune cells. Retroviruses and lentiviruses work perfectly fine, but they randomly integrate their sequence. And that actually, it diminishes the amount of control you have over how you want your new genetic material to interact with the immune cell's endogenous genome and its endogenous set of functionalities. And so to do these targeted knock-ins, again, we just have a DNA template or a CRISPR-Cas9 or Cas12a or whatever version of nuclease you're using that cuts at a desired target site. And then at the same time you electroporate in the nuclease, you also electroporate in a DNA template. And this can be in a variety of formats, either linear double-stranded DNA, linear single-stranded DNA, circular double-stranded DNA, circular single-stranded DNA. All of these work and have advantages and drawbacks for specific applications, but they all have largely the same structure. The new sequence that you want to introduce is in between two what we call homology arms, or regions of DNA that are perfectly matching to the edges, to the 5' and 3' end of the cut site that you had in the genome. If the cell is dividing and is capable of undergoing homology-directed repair, the cell will think that it's it's repairing a double-stranded break that you generated by using an opposite chromosome or a different, its other copy of a gene that was recently synthesized. But instead, you're providing a new copy. You kind of trick the cell into using your new DNA, and it integrates in. It doesn't really know that the DNA in between the homology arms is new. It just goes ahead and integrates it in. And so what this allows you to do is have total sequence control over the final genomic sequence that the cell has. You get to choose exactly where your new sequence goes in, and you get to choose exactly what sequence you add in at that site. Here's just a demonstration of a gene knock-in of a GFB to create a fusion protein at an expressed housekeeping gene RAB11A. But because all we have to do is build a new DNA sequence and have a different guide RNA, but to target essentially anywhere in the genome that we want, you can rapidly go across the genome and integrate new genetic sequences at desired target sites. Here's a series of a bunch of different kind of cytoskeletal or organelle localizing genes that we made targeted GFP fusions with. And so these are live T cells that now have a GFP fusion at actin or at their microtubules or surface receptors or something. And so non-viral gene knock-ins allow you to rapidly go across the genome and introduce new genetic sequences wherever you'd like. And actually, this non-viral approach has been broadly applicable across the hematopoietic lineage. And so you can go across essentially any isolatable, inculturable primary human immune cell, and you can do the same thing, electroporation of a CRISPR-Cas9 component and then a non-viral DNA template can mediate targeted DNA integrations. And that's across different types of T cells, including standard alpha betas, but also gamma deltas and regulatory T cells. You can do this across B cells and K cells. You can also do it at the very root of the tree in hematopoietic stem cells, or even in iPS cells that have been derived from that can differentiate into immune cells as well. So you can choose what cell type you want to integrate this DNA in, and you get total choice of where in that cell's genome you want to integrate new DNA, and that's a lot of power. And so just to show that we can do this in therapeutic settings, I'm not talking about any of the industry clinical trials that now have used this. These non-viral targeted knock-ins for a number of years, but this is an early approach that we did. Just taking because it's so easy to generate these therapies, all you have to do is build the DNA sequence. None of the costly viral vector production and like extremely long time scales to build clinical grade viral vectors. You just need the DNA. And so this is an example of a gene therapy that we've pushed forward at me and colleagues at UCSF, focused on a single family that has a one-of-a-kind autoimmune disease based on a compound heterozygous loss of CD25. Two different mutations, wholly unique to this specific family. Well, we could go into the affected children of this family and actually correct their causative mutation. For the first time in these patients' lives, they have T cells that accurately or faithfully express functional high affinity IL-2 receptor, CD25. And so hoping that this can actually be a gene therapy for by correcting this in these patients' regulatory T cells, actually be a potential therapy for this one-of-a-kind disease. But thinking much more broadly, beyond if just correcting a mutation, because by integrating new genetic material into a specific site in the genome, you actually create a lot of unique advantages in how you can integrate synthetic sequences with existing endogenous components. And that can be by creating new specificity for existing surface receptors, or creating new signaling while keeping the binding properties of a surface receptor constant. You can change expression circuits of an existing product, or you can put a new gene under an existing regulatory control. And these are just a handful of examples of how endogenous you can create more powerful therapeutic possibilities by integrating synthetic DNA with endogenous sequences, because you get to choose exactly in the genome where it goes. So one specific example for T cell and cancer immunotherapy is going into the endogenous T cell receptor and rewiring its antigen specificity. We don't necessarily need to change how the T cell receptor signals. We just want to change what it binds to. And so an example homology-directed repair template to do that goes in, and we can edit at the existing human T cell receptor alpha locus and just put in a new antigen specificity. We also have to put in a new beta chain as well if you're only doing a single editing, but you can do this multiplex too and edit at both the beta and alpha locus at the same time. But we can just change by just changing the antigen binding specificity of a TCR. You can have a smaller gene product that goes in, and you also at the same time you put in a new antigen specificity, whether it's a CAR or a TCR, you knock out or remove the endogenous TCR. And so just to show some old data that we published, and this has now been replicated in humans and clinical trials. But you can generate T cells that are just as, if not more, functional than randomly integrating random integration of a TCR or a CAR using traditional lentiviral approaches. And so just tell you about, since that was all old stuff that we published years ago, just tell you about some new stuff. Again, there's unique advantages that come from being able to choose where in the genome you want to target your cells. And so everything I just showed you was targeting a CAR or a TCR into the exonic regions of the TCR alpha gene to integrate a new sequence being expressed under the endogenous regulatory control of the TCR. But by merely just shifting a couple hundred or kb or so down in the track locus, and instead of knocking our CAR or TCR into an exon, knocking it into an intronic region, again, choosing where this exact same sequence goes in, because you get to you have control over the exact integration site. You create a kind of a unique advantage by integrating into an intronic region. Successful knock-ins of your sequence still result in knockout or loss of the endogenous TCR. You have a new CAR or a new TCR that's sitting smack dab in the middle of the old one, so it disrupts and knocks out the existing one. But crucially, by integrating at by choosing to integrate at an intronic locus, all of the unsuccessfully edited cells, which are largely cells that have gotten a CRISPR-Cas9 cut, but were repaired by non-homologous end joining, a different repair pathway that introduces short indels. Those short indels, if present in an exon, result in gene loss. That's how we do gene knockouts using CRISPR-Cas9 systems, because they introduce a frameshift mutation. But in the intronic region, those short indels are actually spliced out of the final sequence, which means that cells that don't get a successful knock-in end up largely remaining TCR positive. This allows us to do a very simple approach of just depleting or doing a negative selection on CD3 or the endogenous TCR, and we enrich all of our successfully edited cells to close to 90% purity, while also getting rid of the potentially dangerous TCR positive cells that still have some endogenous antigen binding potential. And so we can, by choosing where we integrate this at an intronic region, we enable a touchless negative selection of successfully edited cells, and it didn't cost us any additional DNA at all. We just choose where to integrate it. So I hope there's a couple different vignettes about how being able to have total control over where you integrate DNA into the genome actually ends up having a useful therapeutic benefit. And so next I'll talk about how we've iteratively continuously developed this ability to use these non-viral targeted gene knock-ins to integrate ever larger segments of DNA, which with the kind of the underlying assumption that the more DNA you can integrate, the more power you have to manipulate a cell. So as we first published these non-viral knock-ins in 2018, we've continuously been developing the underlying technology since then. And so while at first these knock-ins were relatively low efficiency compared to traditional viral vectors, we've published a lot of this stuff over time, but iterative development of the underlying technology has allowed us to both increase the efficiency of the integrations, but also you can trade that increased efficiency for introducing ever larger DNA templates. So larger DNA templates integrate at less efficiency than smaller ones, but as we've improved the underlying method, we've been able to slowly increase the overall size of the DNA template that we can introduce. And so just a couple of references kind of along the way of as we've increased the integration size. And so it's now, about a year and a half ago or so, we were able to now push efficient non-viral integrations larger than the packaging capacity of either AAVs or even the most widely used retroviral and lentiviral cassettes. And we've continuously been increasing the underlying methodologies. Bioarchive that we have out now, we have kind of an optimized methodology that we've now been able to integrate over 5 kbs of DNA across a cohort of 24 people. So the 24 human donors T cells, you can see the variable efficiency, but on average about 50% efficiency of those knock-ins of an over a 5 kb template at the track locus. And actually, if we really push the system, we've been able to integrate over 10 kb sequences at efficiencies about 10 to 15% across a number of donors. And so the details are all up on the bioarchive or previous methods that we've put up, but we've now been able to increase our knock-in efficiencies to almost 10,000 base pairs, significantly larger than what you can do with viral vectors. And again, all you got to do is make a plasmid or make a linear double-stranded DNA sequence. So it's much easier to make these templates as well than traditional vectors. And so who knows where this stuff is going? But the point is that over the last couple of years, we've dramatically increased the efficiency of these integrations to about largely the same as traditional viral vector-based approaches, while also being able to integrate larger DNA templates. And that's kind of changed the problem for us when we think about cellular therapy development, which is no longer like, how do we get the DNA into the cell? How do we get a large enough DNA template into the cell? But rather, what in the world do we do with all of the genetic material that we can efficiently add into a cell to make the most effective therapy? If you have 5 to 10,000 base pairs of genetic space, how do you build the right sequences into that genetic landscape to build the most effective drug? And I think the way we've kind of framed this problem is that we're not sure any one single approach is best, but we have lots of ideas about how to improve the efficiency of these cells. So rather than going one by one and just testing, iteratively single sequences that hopefully can improve the efficiency of a T cell in a cancer setting, we said, why don't we just figure out a way to test all of our ideas simultaneously? And so that bounces the problem into how can you run pools or large numbers of DNA integration simultaneously in a pooled population of T cells and actually understand and sequence out selectively what those individual cells do. And so again, we've published the molecular biology underlying all this, but we can do both amplicon-based sequencing of large pools, initially in this first story of dozens to 100 or so DNA sequences tested simultaneously at a time, and we can use amplicon sequencing to read out their unique barcodes. But we can also link these gene expression sequences to these variable synthetic sequences that we're knocking into a T cell. We can link them to single cell RNA or single cell attack readouts because the barcodes that are all expressed within mRNA sequences. So you can pull them down with just off-the-shelf single cell sequencing technologies. So we can set up screening assays or pooled screens, where, and this is an example of an in vivo solid tumor xenograft screen with a pooled library of about 36 different genetic sequences that are being synthetic sequences that are being integrated into the track locus along with a TCR and overexpressed to try to improve the functionality of T cells in this specific environment. So we can both look by traditional abundance-based or amplicon sequencing analysis and see that a variety of constructs in the pool increase in abundance inside the tumor compared to the blood. So it's awesome. But we can drill down much deeper by linking these pooled perturbations with single cell phenotyping. And so we can actually look at the exact phenotypes of all of the cells that got each of the different perturbations. And so this allows us to almost like create a data set that we can do kind of like flow cytometric style gating on different cell populations, but we can essentially like computationally sort on any population that you desire, whether it's surface expression of a certain surface receptor or expression of an entire panel of genes or some computationally defined signature. But we can look within each of these populations of tumor infiltrating cells and see which synthetic sequences enriched or drove cells to acquire that phenotype. So this kind of allows you to go in and say, I want to determine I want a cell to acquire a certain phenotype in this setting. Well, you can then run a big screen and find exactly which constructs drive that phenotype. But then maybe you say, actually, I got the phenotype wrong. I want a slightly different metric, or I want cells that do 2 or 3 or 4 phenotypes at a time. Well, you don't have to run a new screen. You just go back to the same data set and pull out the cells that express your new panel, and you can see which things enriched in there. So we've actually been able to go back iteratively or over multiple times to these same kind of underlying data sets and build up greater understanding as the desired phenotype of a therapeutic cell has changed, or what that metric changes. We can use the same underlying data to predict what types of synthetic genetic manipulations we should make to generate cells that acquire that. So we just last year in Cell, we had kind of an updated or improved version of these pooled knock-in screens that allowed us to a lot of molecular biology improvements, allowed us to drastically increase the scale of these screens to multiple hundreds, to ultimately into thousands of synthetic sequences at a time. And by lowering the cost and the complexity of actually running the screens, we're able to run these screens across multiple different genetic or environmental conditions or different screening assays. And we can build these kind of genotype phenotype maps of synthetic genetic manipulations of human immune cells in CAR or TCR, engineered TCR settings, and see across different environmental conditions which constructs are context dependent, which ones work well in some environments but not others, but then also which constructs work really well across all of them, and those may be some more basal or more powerful manipulations that are a little bit more adaptable to different environments. And so we've done this both in single stimulation assays, done this in a lot of kind of exhaustion-based screening assays, again, both with multiple different TCR and CAR architectures. And again, you can find context-specific hits, things that work well in just CARs or just TCRs. But you can also find things that work well across multiple constructs as well. And so that's where we've looked at these guys now across a variety of different settings. And so the screening assay, again, it could go up to much larger sequences, and we were able to integrate much larger sequences. And so now this is like knock-ins of going up to 8,000 base pairs of a combinatorial library of cells overexpressing 2 different chimeric antigen receptors, along with 2 different transcription factors overexpressed, along with a chimeric antigen receptor, and applied into an T cell exhaustion or tonic stimulation model. And we were actually able to find out of this a huge library of transcription factor combination. We were actually able to find a unique combination of TFs that synergistically interacted with each other, kind of overexpression of both BADF and TFAP4, that only came out of being able to do as highly unexpected, unpredictable. TFAP4 has been very poorly characterized in T cells, not necessarily one that I would I would choose if I only got to choose one factor, and let alone look at the combination of these two was productive. And so these end up being more effective in vitro and in vivo model systems than either on their own or controls. And so again, just highlighting the fact that we can build genetic discovery systems that allow you to rapidly create new large-scale screening libraries of new synthetic sequences to test in primary immune cells across different environmental settings. And you can associate that with high-dimensional single-cell readouts that allow you to build very rich genotype, phenotype data structures to match the right synthetic genetic manipulation with the right therapeutic environment that a cell is going to go into. But with any technology stuff, there's never you're never done. There's always limitations and stuff. And I want to talk about a little bit of some unpublished stuff that we've pushed beyond the screening methods that we've derived before. And so just kind of like to summarize what I've talked about here, we initially were able to generate a non-viral gene knock-in system that allows you to target new DNA across the genome at specific sites. We've optimized that system that allows you to introduce larger sequences, so you can introduce a couple genes at a time across not just T cells, but any culturable immune cell. We've built versions of this that now allow you to do pooled screens, initially up to 100, and then most recently up to about 10 thousands of genetic sequences simultaneously. But again, all of this, everything that we've built, though, only allows you to introduce a new gene and overexpress it. And also, we've been able to only do multiplex, so you can only do 2 genes at a time. And this has been, to a certain sense, somewhat limiting, because there's lots of other work that's been done across the field to identify genes to knock out, or genes to knock down, or domains of genes to add in. And who's to say that it's only 1 or 2 genes that you need to add or manipulate to create the desired therapeutic cell state that you're interested in. And so what we've been trying to do is build a version of these genetic screening system that is truly fully and generalizable, can make complex genetic perturbations of any type, any number of perturbations at large scale, but then combinatorially any of those perturbation types together, and do that all simultaneously in large-scale pooled screening methods. And I think in the method that we've developed, which we call CRISPR-All, I'll try to talk to you quickly just for a couple minutes, and hopefully we'll have some stuff out publicly soon. But this kind of comes actually from looking at how do cancers develop their new functions. They acquire complex genetic changes, and they change the way that they behave in response to their environment and go down for them a productive evolutionary path. But they don't do that just by knocking genes out, or by overexpressing genes, or by building new genes through copy number, through domain combinations and chromosomal translocations. They do all of these at once. And it's the combination, or the many different types of complex genetic perturbations all happening in the same cell that allows cancer to acquire its terrifying new functionalities. And so we've kind of like set ourselves the goal is like, well, can we match cancer's ability to genetically manipulate its cellular function and do the same thing with immune cells? Have the same power to make multiplex, combinatorial, multiple genetic perturbations simultaneously, and screen many hundreds or thousands of those together to find optimal combinations of manipulations to match a cell to a specific function. And so this methodology that we've developed, and I'll kind of like just breeze through the molecular biology and stuff, but kind of tries to synthesize multiple different classes of genetic perturbations, each of which have their own methodologies, or their own kind of bespoke methodologies to generate them, from gene overexpression, gene knockouts, gene knockdowns, adding in synthetic genes, which I just talked about, our own work, adding in domains of genes, especially like CAR signaling domains, and all kinds of other tracking and tracing methodologies. Can we combine them together into a standardized, unified kind of perturbation language that allows you to mix and match these perturbations together while creating unique barcodes for every single sequence, and allowing you to do them all combinatorially together? So this CRISPR-All system that we've developed works through combinatorial interactions of a handful of base modules that encode for full-length genes, whether synthetic or natural, domains of genes, also whether that's coding or non-coding, and then gene knockdowns through shRNA architectures, and gene knockouts through guide RNA-mediated knockouts. And there's a huge amount of molecular biology underneath the hood of this, but what this allows us to do is to actually build a single screening system that can simultaneously screen different perturbation types together in the same assay, or screen any combination of these perturbations together. And so again, just say there is quite a bit of molecular biology work went into building these modules and making sure that they all interact together and all can efficiently edit cells. But these modules are then able to be iteratively built up together one by one, with the modules going on one side and then unique barcodes building up on the other side. That again allows for either amplicon sequencing or any mRNA, off-the-shelf mRNA-based single cell sequencing methodology to now be compatible with any type of genetic perturbation that you want, all in the same assay. So just to show that we can actually do this, here's an example of a CRISPR-All construct that overexpresses two genes, knocks out one gene, and knocks down a final one. There's a huge amount of genetic elements that encode into that, but because we have a standardized architecture or standardized syntax for this, the actual user of the system just has to kind of define what genes do you want and what genes do you want to knock out or knock down. And so what we can see is that of the edited cells here, the vast majority of them have acquired both the encoded gene knockout and the gene knockdown. And actually that and all of this encoded in a single DNA sequence that has a unique barcode that tells you exactly what perturbations were made. We've done this now in much more complex perturbation combinations and stuff. And so I'll just show you kind of like some of the underlying data. This is a syntax of overexpression of 5 genes. Again, that's big. That's a lot of DNA. This is 5 to 6 kb of knock-in, but we can do that pretty efficiently. And all 5 genes end up being successfully expressed in the syntax. But what if you want domains of genes? Well, in this case, we broke up GFP and mCherry into 2 distinct domains, and then combined them back together as if you were combining like signaling domain elements or something like that. Again, knock-in followed by efficient expression, recombination, or reconstitution of both of these genes from their constituent domains. But we can also do gene knockouts by integrating a new gene. We knock in a gene, but then we can also knock out arbitrary genes by including elements within that integrated circuit that codes for each specific gene knockouts. So this is again looking at 2 genes knocked in, gating on the genes that got a knock in. We can see that we have efficient knockout of all 3 of the encoded genes. And then just to reiterate, right after this, there's a barcode that tells you exactly what manipulations happened, even though these knockouts are happening at multiple distinct sites in the genome. We can do this if you don't want to knock out a gene, you just want to knock it down. Well, you can do that too. These are shRNAs encoded here. And we can see that rather than complete knockout, as we can see with these plots, the same 3 genes targeted with shRNA-mediated knockdowns mediate MFI shifts across the entire population, but do not cause overall loss of the gene. So again, you can control gene expression. And finally, all of these can be combined together into individual constructs. So we can overexpress genes, put domains of genes in, while at the same time doing knockdowns and gene knockouts. And all of this has a unique barcode that encodes for it at the end. And so so this CRISPR-All system, which we're now running a whole bunch of fun combinatorial and screens with, has been exciting to develop. And I think it's kind of the next iteration of CRISPR-based genetic perturbation systems, and is somewhat of a unification of diverse bespoke manipulation systems. Just so we can do this in a couple of screening systems as well. We did a screen of 4 of the existing FDA-approved chimeric antigen receptors, Kymriah and Yescarta, and then the BCMA targeters, Abecma and Carvecti. But we ran a screen of those 4 different FDA-approved CAR T cells against a library of 220 proposed gene knockout targets that we and other groups have identified through screening or rational design, it can potentially improve CAR T cell function. And what we found is that we can find some hits. In this case, MED12 came out as the most reproducible hit across all 4 CARs. But we've found large divergence in that you can find gene knockouts that work really well with an individual CAR, but do not work very well at all with others, because of differences in their signaling domains or differences in their binding kinetics. But all of this data, this complex data structure, could be generated in a single screen using this CRISPR-All system. And we're now running much more complex screens like this in the lab. And so, just to reiterate, this CRISPR-All system allows you to test things across perturbation types. And so, one of the more complex screens that we're actively running is a library of essentially what we built is a database of all proposed gene overexpression, gene knockout, gene knockdown, CAR signaling domain, CAR binding domains, synthetic gene overexpressions, all clinically used or late-stage clinical trial CARs or FDA-approved CARs. All of these different things from across the field can be combined into a single screening library that allows you to then rapidly test across whole bunches of different environmental settings and conditions to try to find the optimal genetic manipulations for the specific therapeutic environment that you're seeking to develop drugs in. So this CRISPR-All system is kind of I think of as a unified genetic perturbation language. It kind of builds off of a lot of the themes of how we've genetically manipulated primary human immune cells over the past 6 or 7 years. Just to kind of leave with kind of the way that we approach building these kind of curative cellular medicines is rather than like the traditional approach, it's define a new construct that you're going to make, a new CAR, a new binder, a new signaling domain, some new gene knockout, and then doing a whole bunch of work to examine how that manipulation affects cell function. I think we should be focusing a lot more on defining what is the ideal phenotype of a cell for a specific therapeutic indication. And then we can run very rapidly across all kinds of different perturbation types. We can rapidly determine what synthetic genotypes allow you to get there to that specific perturbation or to that specific phenotype. So define the phenotype and then find whatever works to get you there. And we can rapidly do that through the screening methodologies and pooled screening approach that we've developed. So anyway, I want to thank all the people that have been working with me on that, including my mentor during my clinical residency, Ansu Satpathy, and now all these folks are coming with me into my own lab here at Stanford, but also presented a lot of work that it did previously at UCSF with my PhD mentor, Alex Marson. And so I hope kind of this kind of technology first and genetic manipulation first approach to manipulating the function of primary human immune cells to build better curative cellular medicines across cancer indications has been, it's interesting. And thanks for your time today listening to my presentation.